Ready na? Ready na ba kayo? Thumbs up kung ready na. Ready. Thumbs up. Gamitin niyo yung nod extension. Natutuwa ka dun sa emoji. Nalaman ko lang yan nung isang gabi. Tapos parang, wow, ang cute. Ayan. Nice, nice, nice. Okay, ready na. So, basically, in this subject, we will discuss the central dogma of molecular biology. Has anyone heard of the central dogma of molecular biology? Thumbs up kung narinig nyo na. Thumbs down kung hindi pa. Narinig na ni Ronnie. Okay. May nag-erase ng hand si Gwyneth. Yes, Gwyneth. Narinig mo na ba yung... Wait lang. Ako, hello ma'am, pwede po. Hindi po kasi ako makapasok. Nandito na ba si Shanaya? Shanaya, gun. Class.
Dito po, ma'am.
Wala pa? Ay, nandito.
Wala pa po ako.
Wala? Wait lang. Ang kailangan ata yung USI email address, no? Para makapasok. Dito sa class. Wait lang.
Sige po, send ko na lang po sa messenger.
Okay. Ay, nandito na pala. Okay. Nandito na ba si Bernas? Si Mr. Bernas?
Wala pa ma'am sa G-class ma'am.
Okay. Nandito na pala. So, okay. Nag-chat kasi dun sa chat box. So, nandito na kayo, no? Meron pa bang hindi makapasok? Wala naman. 45 na tayo. So, okay. Continue. So, nasan nga tayo ulit? Central dogma of molecular biology, right? So, in this subject, we will discuss the central dogma of molecular biology sa kabuuan ng first exam. So, the central dogma of molecular biology basically shows us how DNA is transcripted into RNA, and then RNA is translated into proteins. So, yung central dogma, basically, DNA to RNA to proteins. And for this lecture, we will only study the hereditary information. So, wait, sorry, ang gulo ng slide. So, now we will discuss all things related to hereditary information. And as you can see, here is, if any one of you is a K-pop fan, here is Jungkook from the music video DNA, which I played earlier. And may naririnig akong army. So, we will discuss the structure of the DNA in the first part of the lecture, and then we will discuss all about hereditary information in the form of genes. So our objective for this lecture, first, to learn the hereditary information contained in all living things, the DNA structure and how it is packaged in chromosomes, and to study the genes and how changes in the genes affect living things. So first, let's look at the bigger picture. So, what are living things? So, can anyone tell me what are living things? What differs them from non-living things? So, do you have any ideas what differs living things from non-living things? Raise your hands if you want to answer. Sino yung army kanina na ano? Sino yung army kanina? Ano sa tingin nyo? Hello, Arian. So, ano sa tingin mo yung characteristics ng living things?
I think living things are those who are capable of growing and producing their own food or capable of eating for growing.
Sorry, wait. I can't hear you clearly. Ano ulit? Type mo na lang. I-type mo na lang sa chat box.
Living things po. Hello, ma'am. Am I speaking to you right po?
Pwede i-type mo na lang sa chat box, di ko marinig. Sorry, I'm so sorry.
Okay.
Merong parang ah, echo. Ano kaya? Merong mga di nakaka-join sa Gmeet dahil sa internet connection. Um, okay lang, ipo-post natin yung recorded lecture. So, asan na tayo ulit? Living things, di ba? Okay. So, ano nga ba ang living things? Um, Capable to grow, and some are also producing their own food. That's a correct answer. Living things are capable to grow, and they can, some like plants can produce their own food. Um, so um, living things, what differs them from non-living things in the bigger picture is life. So, syempre, living things, life, sobrang simple, di ba? So, for centuries, our ancestors, human beings for centuries, we have been trying to define what life is in a spiritual and or philosophical level. Pero, this is not your religion or philo subject, right? So we are going to look at living things in a molecular level. So can anyone give me an example of a living thing and a non-living thing for comparison lang? You can type on the chat box, or you can raise your hand. Si Arian, type ka ulit ng example ng non-living thing and living thing. Gagamitin lang natin example. Round out. Okay. Hello. Can anyone give me, ayun, uh, non-living thing daw, army bomb. Okay, living thing. Sige, ako na lang magbibigay ng living thing. Okay, BTS daw. BTS, humans, di ba ang BTS? So, living thing sila. Non-living thing, army bomb. Okay, yun yung gagamitin nating example. So, uh, living things, so in this picture, here we look at a cell of a zebrafish, and it is undergoing cell division. And as you can see inside those cells, there is those tiny orange dots which contains the hereditary information for zebrafish. So these cells containing hereditary information for zebrafish will create a zebrafish, obviously. And a zebrafish egg cannot produce a human being. And similarly, human beings cannot produce zebrafishes. And so we now question what are um, in this hereditary information, or parang we now question how this hereditary information makes us all living makes up all living things. So again, living things, they have life, and human beings like Jungkook can reproduce. Human beings can produce another human beings, but non-living things they cannot. So an army bomb cannot produce another army bomb. So we now establish that all living things are made up of cells, and that they contain hereditary information. And you've learned in your previous classes that the nucleus is the part of the cell containing hereditary information. Alam niyo na to lahat, pero refreshment lang. So now, how did scientists know the molecule which contains hereditary information inside the cell? We already know that DNA is the hereditary information uh, con- DNA is what contains the hereditary information inside the cell. But how exactly did scientists know that it is the DNA? So let's take a look at this diagram. So focus on the left side diagram first. So here we have an S strain of bacteria, let's call it red bacteria, and they are pathogenic, meaning infectious. And then this red strain of bacteria mutated to become the R strain, which is non-pathogenic. And what scientists did is that they heat killed this S strain, they killed it, okay, and then they placed it in a petri dish with the yellow bacteria and they cultured it. So the bacteria now grew right in a culture. So what surprised them is that yung nag-grow red bacteria. So remember, yung red bacteria pinatay nila. Pero tapos nilagay nila do'n yung yellow bacteria. So, red bacteria plus alive yellow bacteria, yung nag-grow red bacteria. And them, na-curious sila. And then they concluded that the molecules that carry hereditary information are present in the S strain cells. So, the red bacteria, the hereditary information inside the red bacteria are still present, and then the yellow bacteria, they gathered or they acquired the hereditary information from the red bacteria, even though they are already dead. So that happened in 1920s. So in 1940 naman, here, let's look at the right diagram. So they were looking at the molecules, or they were trying to identify which molecule inside the cell contains the hereditary information. So again, they had this red bacteria or the S strain, and they fractionated it. So fractionation is a way to extract molecules inside the cell and to um, purify the molecules, or parang i-separate sila from each other. So they purified RNA, they purified proteins, they purified DNA, they purified lipids, and they purified carbohydrates. And similarly, from the first experiment, they placed the yellow bacteria in each plate containing each of these molecules. And they observed that only DNA gave rise to the red cells, para or parang yung na-observe nila, yung ibang molecules na na-separate nila, yellow bacteria pa rin yung nag-grow. Only the DNA gave rise to the red cells. And so they concluded now that DNA carried the hereditary information. So basically, kinumpare nila lahat ng molecules inside the cell, and then they found out that DNA is what contains the hereditary information. And now, they in this time period in 1940s, they still don't know how DNA or the mechanism behind how DNA passes on its hereditary information, or how it is being carried from one cell to another. So, see, Rosalind Franklin now, she's a scientist. In 1951, she photographed the DNA using X-ray crystallography to study the DNA structure. So she's a pioneer scientist of the DNA structure. So Rosalind Franklin, she photographed the DNA using X-ray crystallography. And then in 1953, nakita ni James Watson and Francis Crick yung notes ni Madam Rosalind Franklin. So wow, nakita nila yung notes ni Rosalind Franklin, and they said to themselves that we can mag-build daw sila nung structure ng DNA. And so they did that in 1953. And all of you may have already know about them. And they won the Nobel Prize for Physiology and Medicine in 1962. And yung sad story dito is that Rosalind Franklin did not win the Nobel Prize because she died before the Nobel Prize was awarded. And that's kind of unfair because there's a rule in Nobel Prize that if you die, you won't be awarded of the prize. But she's actually the pioneer for the DNA structure, and that's what happened. So DNA structure. So in this lecture, we are on the first part of the lecture, we are going to look at the DNA structure. So let's dive deep into it. So nucleotides. Um, these are the molecular building block of DNA. They are the basic unit of DNA. So they are composed of a sugar, itong blue, a base, itong green, phosphate, and a phosphate, itong yellow. So this is just the simplified version. So sugar plus phosphate plus base. So alam niyo na 'to pero review lang. Review lang natin para sa next lectures natin refresh kayo. Okay? So first, let's discuss the sugar. So in your biochemistry class, you've studied different kinds of sugar molecules, right? So in DNA, we use the 5-carbon sugar called pentose. So there are 5 carbons, right? 1, 2, 3, 4, 5 here. So when you read this one, there's a prime mark, right? So if you read this carbon, this is called the 5' carbon. And there are two types of sugars in nucleic acids. This is the ribose and this is the deoxyribose. And the difference is that the ribose has a hydroxyl group in its second carbon. And but what we are going to use in what is used in DNA is the deoxyribose, this sugar right here. And then the bases. So you've already been taught this, but I will just refresh you. So bases are nitrogen-containing ring compounds, either pyrimidines or purines. So pyrimidines are one-ringed, and they are cytosine, uracil, and thymine. And purines are two-ringed. They are adenine and guanine. So you can make a table like this to remember them, but you can also use the mnemonic, I cut the pyramid. So this is the mnemonic that I use. So I or one ring, cut, cytosine, uracil, and thymine, the pyramid, pyrimidine. Okay, that's how I remember it. In DNA, we use AGCT. These are the bases that we use for DNA. Okay. So, masyado ba akong mabilis? Sorry. So, um, now we have sugar and base. So, when you combine them, um, sugar and base is combined by sugar and base is called the nucleoside. So, remember, if there is only the sugar molecule and the base, um, you will only have a nucleoside. So what makes it as the nucleotide? A nucleotide has a phosphate group. So the nucleotide has a phosphate group which is attached to the carbon 5 hydroxyl of the sugar. Okay, so now we have established the structure of the nucleotide. And again, this is the basic unit of the DNA. And again, nucleotide contains a nitrogen-containing base, a 5-carbon sugar, and a phosphate group. And this is just a representation of how it looks like. So, okay, so look at it, okay? Okay, hinga muna. Yan yung structure ng nucleotide. So, can anyone tell me what are these structures? What is the one on the left and the one on the right? Please raise your hand. Review lang. Ano to? Ronnie, yes.
Yung nasa left po, nucleoside. Yung nasa right po, nucleotide.
Tama. So, itong nasa left ay nucleoside dahil merong sugar and base lang. But nucleotide, remember, has a phosphate group. Okay, alam nyo na. So, here now, find the nucleotide. Kung sino makahanap, you can raise your hand. Find the nucleotide. Balikan natin si Jungkook. Review lang to. Bigyan ko kayo ng 10 seconds. 9, 8, 7, 9, 1. May nakahanap ba? Sirit na? To. Ay, sino? Sino? Sorry, napindot ko na. Napindot ko na. Sorry, Nicole. Okay, next time na lang, Nicole. So, ito yung nucleotide. So, bakit? Andyan yung sugar, andyan yung base, andyan yung phosphate group. So, next time na manunod kayo ng music video ng DNA, makikita nyo na yan. So, ito, nucleotide ba to? Hindi. Hindi yan nucleotide. Bases lang yan. Kasi, ito, two rings. So, yung two rings, remember again, they are purines. And one ring is pyrimidine. Okay. Ito rin. So, as you can see here, these are bases. Pero dito, kita yung sugar, right? So, yun. Yun lang yun. So, now, we look at how a DNA strand is made. So, So you have, sorry. So you have one nucleotide and another nucleotide, right? Because they are the basic structure of the DNA. So how do they connect? So here, you can see that the lower nucleotide is connected at the 3' of 3' carbon of the upper nucleotide. So, a nucleotide is always added at the 3' end of a chain. So, when you look at the DNA strand, it always starts from the 5' to the 3' end. So, DNA strand always grows from the 5' to the 3' end. So, you just read this from left to right. So when you read this is just GCAT. Okay. Remember always that a nucleotide is added here at the 3'. Okay. So, wait lang, balik tayo dito. So, DNA strand pa lang, di ba? So, now, naalala nyo, yung structure na na-discover ni Watson and Crick, double-stranded yun, di ba? So, how does it become a double-stranded DNA? So, let's look at Chargaff's rule. So, in Chargaff's rule, it says that the amount of purines is equal to the amount of pyrimidines, and the amount of adenine is equal to the amount of thymine, and also the amount of guanine is equal to the amount of cytosine. And as you can see here, adenine is bound to thymine, and guanine is bound to cytosine. And that is always the case for the bases. So when you connect the bases, ganyan parati kung paano sila mag-bind. And in Chargaff's rule, ito yung rule na ginamit actually ni Watson and Crick to establish the DNA structure, the twisted ladder structure of the DNA. So they alam nila na ang amount ng ang amount ng purines, adenine and guanine ay always equal to the amount of pyrimidines, which is thymine and cytosine. So these bases are bonded by hydrogen bonds, and they are now called the base pairs. So this is one base pair, this is another base pair, and adenine and thymine always have two hydrogen bonds between them, while guanine and cytosine have three hydrogen bonds between them. So sa tingin nyo, alin yung mas malakas sa dalawa? Raise your hand. Meron ba? Okay. So, yung mas malakas sa dalawa ay yung sorry, merong left. Mahina yung internet nila. So, itong guanine and cytosine because they obviously have three hydrogen bonds. So, yun lang yung base pairs. Okay. Wait. Now we have the DNA structure. It is a twisted ladder of complementary strands which are anti-parallel to each other. So let's define each um, term used to describe the DNA structure. So twisted ladder, as you can see, it is a twisted ladder, right? Yung nasa right. So bakit kaya twisted ladder ang DNA? Twisted ladder ang DNA kasi there is a problem in how to package it inside the nucleus. So if you stretch a DNA inside one cell, it will actually um, uh, will be 2 meters long. So imagine niyo, you have millions of cells inside your body, right? So if you stretch each DNA, 2 meters yon. And if you connect all those DNA strands, pwede kang makarating ng moon ng pabalik-balik or ng sun ng pabalik-balik. Ganun kahaba yung DNA. So twisted ladder siya for it to be packaged inside the cell, inside the nucleus. So okay, Complement complementary strands. So, why are they complementary strands? So, 'di ba nakita nyo kanina merong base pairs? So, these base pairs are complementary to each other. So, yun lang naman yun. And then they are anti-parallel to each other because they run opposite from one another. So as you can see here, 5 prime to 3 prime, 5 prime to 3 prime, and we will discover in our lecture next time in DNA replication that this is the 3 prime to 5 prime strand, and this is the 5 prime to 3 prime strand, and it will have a significance on in DNA replication. So, ganun lang, pabaliktad lang sila, kaya tinawag na anti-parallel. Okay. No. Oh, nakita niyo yung sagot. So, yan, existential crisis. Bakit ba tayo nag-aaral ng structure ng DNA? Bakit kaya? Bakit ko kaya 'to inaaral, 'di ba? May ano bang kahalagahan nito sa buhay ko? So, the structure of the DNA provides a mechanism for heredity. So, bakit yung slides ko nagkaganon-ganon? So, when before the time of Rosalind Franklin and Watson and Crick, 'di ba? You remember um, people or scientists know that DNA contains the hereditary information, but they don't know how DNA replicates or the mechanism behind it. So when Rosalind Franklin, Watson and Crick discovered the DNA structure, it answered all those problems. So because now scientists can see how DNA replication happens. So you see, the DNA is a double strand, right? And if you use either of the template or either of the strand as a template, you will get the same strand. So first the parental DNA, if you use the S strand as a template, you will you will still have the S prime strand, and vice versa. Also for the if you use the S prime strand as a template, you will still have or you will still get the S strand. So that's what it means for yun yung meaning ko bakit complementary yung DNA. Kasi kahit anong strand dun sa DNA, gamitin mo, makukuha mo pa rin yung kabilang strand to make a new DNA. Okay. Now we discuss the genes. These are the segments of the DNA that carry the hereditary information. So, you now have an idea of the structure of the DNA, and that that it carries hereditary information. But the units of or there are specific segments in the DNA which actually carry the hereditary information. So there are different segments in the DNA called the genes, which carries the hereditary information. So gene codes for proteins and sometimes RNA. And there is always one gene from for one protein. So, for example, gene A, it produces the protein A, and gene B produces the protein B, and of course, gene C produces the protein C. So, one gene, one protein. Okay. And for you to appreciate this, let's have an example of a gene. Wait lang. Ito, example natin yung human beta-globin gene. So, wag kayong ma-overwhelm dito. Ito lang yung nucleotide sequence ng human beta-globin gene. Kapag sinequence mo tong human beta-globin gene, ito yung ano niya, ito yung sequence na, yung nucleotide sequence niya. Binasa mo lang. So, what is the human beta-globin gene? Diba sabi natin, one gene for one protein. So, the human beta-globin gene, it produces the beta-globin of the hemoglobin, the subunit. So, 'di ba tinuro sa inyo na yung hemoglobin uh, there are subunits, so the alpha and the beta. So yung human beta globin gene, they produce the protein for the beta subunit, itong pink and itong green. And this is important for carrying the oxygen in the blood. Okay? And ito yung nucleotide sequence niya. And yun. Yun lang yung gusto kong i-emphasize na for one gene, there is one protein. Okay. Now, the genome is the collection of all genetic material of an organism. So, all of these genes that you have, all of your DNA, yung collection nun, tawag doon genome. So, analogous siya sa library because a library is the collection of books, right? Collection of information. And these information in the DNA are used to create you or for the cellular or for your cells to function. So, the genome, again, is the collection of, wait, the genome is the collection of all the genetic information. So, a bacteria has a circular DNA. Ito, E. coli, ito yung DNA ng E. coli. So, circular siya, right? And, yan na yung buong genome niya kasi lahat na ng genes ng bacteria, nandyan na. But humans, we have the so-called chromosomes. And these chromosomes contain all of our DNA. And here is a picture of my actual chromosomes from the karyotyping activity in my graduate studies. So I karyotyped my own own chromosomes. Ito yung lumabas. So, normal naman. Tapos yan, XX, diba? So, yan lang. Masaya mag-karyotyping. Yun. So, na-discuss yun na to sa cytogenetics. Pero, yun. So, this now shows us how DNA is packaged inside the eukaryotic cells. So, tingnan nyo, di ba? Yung bacteria, yun na yun. Bilog lang. But eukaryotic cells, humans, we have lots of genes. So, there are lots of genetic information for humans. So, para mapagkasya yon, yung lahat, diba, sabi ko, 2 meters kada DNA pag stretch. Kapag para mapagkasya lahat yon, we have the chromosomes. And let's look at this, how DNA is packaged in eukaryotic cells. And as I've said before, 'di ba, DNA kapag na-stretch mo siya sobrang haba. So, analogo siya sa yarn na loose. So, anong gagawin mo para yung yarn ma-package or package mo, 'di ba? Obviously, iba-bundle mo. So, ito, analogous ito sa chromosomes natin. Kasi dun sa chromosomes nakapackage yung DNA. So, again, human chromosomes, there are 23 pairs of chromosomes, 22 are autosomes, and one pair of sex chromosomes. And you've already studied this in your cytogenetics. So the the chromosome is a structure composed of a very long DNA molecule and associated proteins that carry the hereditary information of an organism. So let's discuss this, diba? Words, parang Wow, ma'am, words, yun lang. Pero yun, di-discuss natin para maintindihan nyo kung anong meaning ng chromosome. So, uh, wait lang, ang gulo ng slide. So, first, let's look at the chromatin. So, the chromatin, um, it is a complex of DNA, histones, and non-histone proteins. So, this is the material which chromosomes are made. So, as you can see, the orange thread is the DNA. And the DNA, it coils around these proteins called the histones. And just like um, these non-histone proteins are attached to the fiber. So, the function of the non-histone proteins, I will discuss it in our later lectures. But remember nyo lang na yung chromatin, basically made up of the histone protein, in which the DNA is coiled around it, and the non-histone protein. Okay. So, para snippet lang, yung non-histone proteins, ano siya, excuse me, meron siyang function sa DNA expression. Pero, yun. I-discuss natin yun later part. So, ayun. So, yun yung chromatin. So, DNA bound to histone and non-histone protein. So, nung 1974, nalaman nila ay parang pinitsura nila yung chromatin. Tapos ito yung picture nila, 'di ba? Parang bead, parang yung sa rosary. So, nakita nila yung nucleosomes. And nucleosomes are the basic unit of eukaryotic chromosome structure. So, it is made up of protein and the DNA complex. So, as you can see here, 'di ba? Wait, dito tayo sa figure na to. Yan. Yung ano, wait. So, ito yung chromatin fiber, right? So, when you zoom in, here are the nucleosomes. Okay? And yun, pinapakita niya yung histone proteins. And when you zoom in further, that's the DNA. So, yun yung na-discover nila noong 1974. Okay. And in 1997, wait. In 1997 naman, they now found out the specific structure of the nucleosome. So here is the core histone forming an octamer. So these proteins are very small. So yung proteins na yon, nagpo-form sila ng octamer in which the DNA is bound. So the histone octamer plus the histone plus the string of DNA will make up the nucleosomes. And the nucleosomes are coiled, parang magko-coil sila ng magko-coil, parang continuous coil lang. And this will make beads on a string nucleosome array. Yun yung itsura nila. So, imagine nyo lang, parang yung sa rosary, pero nakapulupot yung DNA doon. So, ayun. So, ito, ano to? Nakita nyo na. Well, so, this is the structural representation of the nucleosome. So, di ba sabi ko nung una, kapag may nakikita kayong structure na ganito, parang protein yan. So, yan yung histone, yung representation ng histone. Yung picture na ganyan, pinapakita yung atoms. Ang tawag dito sa model na to ay space field model. So, the colored ones are the histone proteins, and specifically, these are called as the H2A, H2B, H3, and H4. Yun yung histone proteins, and they are very small. And remember that DNA is coiled around these proteins. So, ngayon, yung nakikita niyo yung gray structure na nakapulupot doon sa colored proteins, yung colored ones, yan ngayon yung DNA. So, yan lang yung representation ng nucleosome. So, Again, ngayon, pag pinakita ko na sa inyo tong picture na to, maiintindihan nyo na. Sorry. Um, wait lang. May distraction. Okay. Diniscuss natin yung DNA structure, right? And diniscuss natin na all living things are made up of cells. And the hereditary information is found inside the nucleus. And the nucleus, inside the nucleus are the chromosomes. And the chromosomes solves how DNA is packaged, because if you stretch the DNA ulit, it is too long. So the chromosomes solves it, parang yung yarn. And if you zoom in further, you will see the chromatin. They are fibers which are composed of the histones. And histones are the units of the chromatin. And nucleosomes, I'm sorry. When you zoom in further on the chromatin, you will see the nucleosomes. And the nucleosomes, they are composed of the histone proteins and the DNA wrapped around it. And when you zoom in further, you will see the DNA. And the DNA has the basic, uh, the nucleotide is its basic unit. And the nucleotides have different base pairs, the guanine, the cytosine, the adenine, and the thymine. And sections of the DNA are called the genes. And genes, they carry hereditary information. Okay. Yun lang yung kabuuan ng diniscuss natin sa first part. So, before I proceed with the lecture, are there any questions about this first part of the lecture? Kung may questions, raise your hand. Kung walang questions, thumbs down. Gamitin nyo yung nod, yung emoji. Kung walang nod, okay lang dun sa raise hand sa Google Meet. May questions ba kayo? Gusto nyo bang, meron bang part dyan na gusto nyo bang i-elaborate ko or something? Okay. May gumagamit ba? Mag-thumbs up kayo kapag na-gets nyo naman. Yun. Yay! May gumagamit ng nod. Okay. Yun. Ha! Yay! Yun. Gamitin niyo yung nod. Cute-cute ng nod. Feeling ko nasa V-Live ako. Alam niyo ba yung V-Live? Doon yung mga K-pop. Yung mga K-pop fans. Ganun. Nag-V-Live tayo. Mga K-pop fans lang makakarelate. Okay. Wala namang tanong dito. Okay. Okay. Hinga tayo. Sana tayo. Yan. Bakit pa? Ayan, mag-existential crisis na naman tayo. Wait lang. Why am I studying the hereditary information? What for? Parang, ma'am, medtech ako. Bakit ko to kailangan aralin? Bakit ko kailangan aralin lahat yung DNA structure? Bakit ko kailangan aralin kung ano yung genes? Ganon. Bakit ko kailangan aralin yung chromosomes? Because you have to understand how some diseases, specifically genetic disorders, occur. Okay, medical technologists, right, in the field of medicine. So we have to use the learnings that we have in all of our subjects to apply for our medical career. So here we will have a case study of two diseases, the sickle cell disease and the beta thalassemia. So, para ma-appreciate yung mga inaaral nyo, hindi lang yung i-memorize nyo kung ano yung structure ng DNA, di ba? So, yun. Sickle cell disease. So, the sickle cell disease is an inherited blood disorder in which two copies of the HBB gene is mutated. The normal blood looks like this normal disc shape, right? But the sickle cell, if you have a sickle cell disease, your red blood cells will look like a sickle. So deformed siya. And this affects the transport of oxygen. And again, this is due to the mutation of the HBB gene in which you inherited two abnormal copies of the HBB gene. So, 'di ba, nakita nyo na yung gene, they they carry the hereditary information. So, also remember in your cytogenetics that you inherit one copy of your chromosomes from your mom and one copy of your chromosomes from your dad, right? Remember nyo, remember. So, remember nyo. Um, Now, in sickle cell disease, you inherited two abnormal copies of the HBB gene. So, yung copy mo sa mom mo and sa dad mo, abnormal pareho. May mutation dun sa HBB gene. Which makes it, which now gives you or like affects the structure of your red blood cells and it causes the sickle cell disease. Okay? Next disorder, the beta thalassemia. So, it is a mutation again in the HBB gene, but this time, the production of the protein beta globin is reduced. So, if you don't have enough beta globin proteins, you won't also have enough um, proteins to make the hemoglobin. And this affects the production of red blood cells, leading to shortage of the red blood cells. So again, the HBB gene, they produce the proteins in the or called the beta globin proteins, itong subunits ng hemoglobin. 'Di ba ito yung hemoglobin? Ito yung beta subunits. So yung production daw ng proteins na to mababa kasi merong mutation dun sa HBB gene mo. Mababa ngayon yung production mo ng proteins na to. And then this affects now the production of hemoglobin and results to the shortage of mature red blood cells. And yun. So, yun yung dalawang diseases na um, parang pwede nyong ma-relate kung bakit nyo inaaral yung hereditary information. So, first is the sickle cell disease, and if you look at it at the genetic level, yung HBB gene yung problema, you inherited two abnormal copies, and in beta thalassemia, yung HBB gene naman ngayon ay mutated and naapektuhan yung production ng proteins, ng beta globin proteins. Kasi, di ba, alalahanin nyo, sabi ko kanina, 'di ba, one gene is equals to one protein. So, yung gene sa inuulit ko lang, yung HBB gene, nagpo-produce siya ng beta globin proteins kasi one gene is equals to one protein. And ngayon, dahil may mutation sa HBB gene, decreased yung production ngayon ng protein na to. And decreased din yung production ng hemoglobin. And decreased din yung production ng red blood cells. And now, it affects how the blood carries oxygen. Mas konti na lang yung oxygen na nakikarry. Okay. So, genetic mutations are a permanent alteration in the DNA sequence of a gene. And there are two types of genetic mutations. Ito yung parang broad type, hereditary mutations in which you inherit them from your parents, and these mutations are present na sa parents nyo and you just inherited them. Yun. And the other one is acquired or somatic mutations, which only occurs during a person's lifetime. So, di mo siya na-inherit. Parang nag-mutate lang bigla inside your your DNA. Nagkaroon ng mutation. Okay. So, yun yung dalawang broad types ng genetic mutations. So, meron pang mas specific types ng mutations, pero later parts natin sila i-discuss para ma-relate. Parang ano lang to, parang refreshments and introductions para sa next lectures nyo, may ano kayo, na-refresh kayo. Okay? So, are all mutations bad? So, existential crisis ulit. Meron bang may idea kung lahat ng mutations bad? Anong ano nyo dito, opinion? Can anyone tell me, are all mutations bad? Thumbs up kung mutation bad, thumbs down kung hindi lahat ng mutations bad. So, nag-agree ba kayo na mutation ay bad? Thumbs up kayo. Sabi ni Ronnie, hindi daw. Paano yung iba? Hindi rin daw, sabi ni Zia, si Rian. Okay. Si Isaiah, si Alvin, si Maria, si Arian. Para akong nagsha-shoutout, no? Okay. Ayun. Shout out sa mga nag-thumbs down. Okay. Sabi nila, di raw lahat ng mutations masama. Tama. Tama kayo. Di lahat ng mutations masama. Pero di ba dito, parang yung sa unang part, parang inaral natin, saan na yun? Iba, nagkaroon ka ng sakit, yung beta thalassemia to si sickle cell, pero mutation pa rin sila. Pero ito. Hindi lahat ng mutations ay bad. So, no. Some mutations in the gene are actually beneficial. Tingnan natin. So, balik tayo sa tree of life, di ba? Sabi natin, life. Life yung uh, characteristics ng living things. Sorry, nag-ano na ako. So, ito yung tree of life, right? And variations in genetic information occurs throughout the tree of life. And we came from a common ancestor. And we evolved from a common ancestor. So, evolution, right? So, kung hindi kayo naniniwala sa evolution, maniwala kayo dahil nasa biology class tayo. So, we all came from a common ancestor. So, dapat pare-pareho yung genes natin or yung hereditary information. Pero bakit hindi? Bakit di natin kamukha yung aso natin? Bakit di natin kamukha yung pusa natin? Bakit di natin kamukha yung ibon? Bakit ganun, di ba? Naisip niyo ba? Yun, di ba? Kung galing lang tayo sa common ancestors. Kasi, nga, merong genetic variation through mutation. So, mutation dito, mutation dyan, mutation kayong lahat. Nagkaroon ng mutations to create different types of living organisms, right? And it created variations in all living organisms. So, new genes are generated from pre-existing genes. So, these are the different types of mutation. So, first is intragenic mutation. So, an existing gene can be modified by mutation in its DNA sequence. So, number one, yan, intragenic mutation. So, ang example dyan, ba't di nakita? Ayan. Ang example dyan ay single nucleotide polymorphisms. Narinig nyo na ata to, pero single nucleotide polymorphisms, basically, dun sa DNA sequence mo, merong yung isang nucleotide doon nagbago. Parang from A, naging G. Diba parang gusto ko ng A, pero naging G. Ganun. So, may single nucleotide doon na nagbago. From A to G or from A to T, from A to T, ganun. Single lang. So, yun yung example ng introgenic mutation. Within the gene, merong nagbago or merong nagmutate. Okay. So, next one is gene duplication. So, gene duplication, an existing gene can be duplicated so as to create a pair of closely related genes within a single cell. So, example natin dito ay gene families. Kaya merong gene families kung nabasa nyo na ito. So, example yung globin genes, right? Diba, naalala nyo sa hemoglobin, merong different subunits, yung alpha and beta, different subunits, right? So, they all belong to one family of the globin genes. Isang family lang yun, yung alpha and beta subunits of the hemoglobin. Under lang sila ng isang family ng globin genes. And nangyari yun dahil nagkaroon ng genetic duplication noong unang panahon. So, the third one is segment shuffling, wherein two or more existing genes can be broken and rejoined to make a hybrid gene, consisting of DNA segments that originally belong to separate genes. So, example natin dito ay yung beta-lactamase genes in antibiotic-resistant bacteria. Narinig niyo na yun, no? Yung BLA genes, yung beta-lactamase genes. So, yung sa bacteria, merong beta-lactamase genes and nagsha-shuffle sila. Yun yung example sa segment shuffling. And lastly, the fourth one is horizontal or intercellular transfer. So, a piece of a DNA can be transferred from the genome of one cell to that of another. So, even that of another species. So, this process is in contrast with the vertical transfer of the genetic information, di ba? Kasi, wait lang. Horizontal transfer, meron kang organism A and meron kang organism B. And ngayon, si organism B, tingnan nyo, bigla niyang nakuha itong gene ng organism A. And that is what is horizontal transfer is. In contrast to vertical transfer is from parents to offspring. So, related sila kapag vertical transfer. Pero kapag horizontal transfer, you got it from another organism. And this is always observed in bacteria. Okay? So, always observed to in bacteria. Kaya nagkaroon ng antibiotic resistance, di ba? Yung BLA gene, yung beta-lactamase gene. So, narinig niyo na ba yung beta-lactamase genes? Thumbs up kung oo. Oo na, no? Yan, na-discuss na yun. Or narinig niyo na, parang familiar, di ba? Beta-lactamase genes. Sila yung nagpo-produce ng enzyme para magkaroon ng antibiotic resistance yung bacteria. And, ano pa ba? Di ko pa pala sa inyo nasabi. So, kapag nakita nyo, italicized yung name, ibig sabihin, gene yan. So, kapag magbabasa kayo ng journal articles, tapos merong italicized, ibig sabihin, gene yon. And kapag hindi italicized, protein yon. So, ito. So, kapag magbabasa kayo ngayon ng journal articles para sa thesis nyo, ganun, kapag italicized, meaning nun, gene. Okay? Yun lang yun. So, now, genetic information is conserved since the beginning of life. So, di ba sabi natin, galing tayo sa isang common ancestor and merong variations. Pero kahit na may variations tayo, meron pa rin tayong parts sa hereditary information natin or meron tayong parts sa DNA natin na conserved. So, ibig sabihin, parang common siya sa lahat ng organism. So, example nun is yung 16S rRNA. Itong 16S rRNA, part siya ng ribosome na makikita natin in the later parts of our lectures, in the future lectures. So, example lang, para lang may example tayo ng conserved genetic information. So, ngayon, yung gene na to, binasa nila, sinequence nila, and nakita nila in humans and in Methanococcus and in E. coli and another human again, similar tong gene na to. So, pareho lang. So, conserved, ibig sabihin, kahit na meron tayong variations, meron pa rin tayong parts sa DNA natin na similar sa ibang organisms. So, yun. And these variations now, for example, they created different blood types. Kaya tayo merong iba-ibang blood types kasi meron tayong variations sa genes. Kaya yung iba sa atin, blood type A, yung iba blood type B, A, B, O. Kasi meron tayong variation sa genes. And another example is that in lactose intolerance. So, noong unang panahon, lactose intolerant tayo. So, kilala niyo ba to? Thumbs up kung kilala niyo to. Alam ko may nanonood sa inyo ng friends. Yeah. Binasa ko yung mga forms na merong nanonood dito ng friends. So, si Joey to. So, dun sa friends, si Joey daw, kaya niya daw uminom ng 1 gallon ng milk for 10 seconds. So, tingnan niyo si Phoebe, tinatimeran niya si Joey. Pero tingnan niyo, natatapon lang naman yung gatas. Ayan. So, lactose intolerance. Example siya ng mutation na beneficial to us. Kasi dati, lactose intolerant tayo lahat. But then, something inside us mutated to create the enzyme to process dairy products. So now we can drink milk or eat cheese or other dairy products. Ayun. And another example of beneficial mutation, hindi sa atin, pero sa bacteria. So, hindi to beneficial sa atin, but antibiotic resistance are caused by genetic mutation. So, as you can see here, the antibiotics are susceptible, right? The bacteria, susceptible pa siya dito. Pero dito, resistant na. After some time, naging resistant na. And beneficial yung mutation na antibiotic resistance sa bacteria. Kasi ibig sabihin, mas mabubuhay sila. Pero sa atin, hindi siya beneficial. Okay? And another is interesting part about mutation is that in sickle cell disease, narinig yun na ata to, di ba? Parang merong protective advantage yung mga tao na may sickle cell disease against malaria kasi hindi naman makakapasok yung plasmodium sa RBC mo kapag deformed yung RBC mo, di ba? Ayun. Saan na tayo? So, protective advantage. So, to ayun na yung end ng lecture natin. So, to summarize everything here. So, living things are composed of cells which contain hereditary information in the form of DNA. And we also learned that the DNA structure and how it is packaged in chromosome. And studying DNA can shed light to hereditary disorders and gene mutation. So, kaya natin inaaral ito, structure ng DNA, itong chromosomes, kasi para malaman natin yung deeper explanation for diseases and mutations. And not all mutations in the genes are bad. In fact, variations in the genes are created through mutations. So, are there any questions about the lecture? Thumbs up kung na-get nyo naman or thumbs down kung walang, raise your hand kapag may question. May question ba? Raise your hand. Review lang, no? Parang most ng mga nang nandito sa lecture 1, review lang to nung lessons nyo sa cytogenetics and sa biochemistry. Parang refreshments lang. Okay. So, now na meron na alam na natin, meron na tayong background sa structure ng DNA and how genes work, we can proceed to discuss the central dogma of molecular biology in greater detail in our next session. So, next lecture, we will cover or we will discuss the DNA replication. So, di ba ngayong lecture, diniscuss natin itong DNA structure and yung genes. And now, na-refresh na kayo tungkol dito. Na-refresh na kayo tungkol dito. Didiscuss na natin yung DNA replication in greater detail in the molecular detail. So, so sa buong lecture 1, sa buong exam 1, sa buong first part ng class natin, ito lang yung i-discuss natin, the central dogma of molecular biology, but in greater detail. And I will again give you examples ng diseases na ma-relate nyo sa central dogma para alam nyo yung mechanisms behind these diseases, and you can use them in your medical career. Okay. So, if wala ng questions, we will end the lecture here. But before you go, wait. May questions ba? May questions? 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 Okay. Wait lang. Meron pa ba dito hindi nakapasok sa Google Meet? May hindi pa ba Google Meet tuloy? Sa Google Class? May di ba nakapasok sa Google Class? Wait. Oh. Wait, so kung wala na kayong questions, I will send you a form, another Google form. And you have to submit it to me before on Wednesday next week at 8 p.m. before 8 p.m. next Wednesday next week. So, meron lang tong dalawang question. Kung nakapagtanong na kayo sa friends nyo from the other section, ayun. Ito, yung Google form. Wait. So, yung form, meron yung dalawang questions. Para ma-assess ko kung meron kayong natutunan or kung meron kayong questions na nahihiya kayong itanong or sorry. Sorry. Oh my God. Sinisipon na ako, guys. Kung meron kayong tanong, pwede niyong wait lang, kaya ko pa. Don't worry. Kung meron kayong tanong, meron dyan sa form na dalawang questions. Yung number 1, kung ano yung natutunan nyo. And yung number 2, kung kung ano yung questions nyo dun sa lecture natin. And yun, we will end the meeting here. We'll end the meeting here. Thank you so much for listening to me. Nalimutan ko. Meron tayong quiz sa lecture 1. Next meeting sa lecture schedule natin Friday ulit. Quiz lang, short quiz para ma-refresh kayo. Okay? So yun pa. Ano pa ba? Wait lang. Tipin ko pa baka may announcement pa ako.
Friday, ma'am? Or...
Yes po. Sa Friday.
Okay.
Ayan. Yun. So, yung mga wala pa sa Google Classroom, sabihan niyo ako. And yung form na sinend ko, i-fill out nyo. 2 lang yung questions doon. Para yon sa class participation. So, para meron kayong grade sa class participation, kailangan yung sagutan yung 2 questions na yon. Simple lang yung question. So, what did you learn? And ano yung questions nyo sa lecture na to? And lastly, meron tayong quiz next meeting, Friday. Okay, yun lang. So, we will end the session here. You can have the remaining time to answer the Google form that I sent, and you have to accomplish it before Wednesday, 8 p.m. Okay? Thank you. Bye-bye.
Thank you po, ma'am.
Thank you po.
Thank you po, ma'am.
Bye. Stop recording ko na. Okay.